Okay, so we hit 11.4, 11.5, cellular response and signal amplification. First slide, so response. So we have the third step in cell signaling. We know the first two steps are reception and then transduction. This stage is where finally the transduced signal actually produces a cellular response. The response leads to regulation of transcription or could lead to other cytoplasmic activities. This stage always occurs in the nucleus or the cytoplasm. Most pathways result in regulation of protein synthesis. The final molecule of the pathway can be a transcription factor, which is something that stimulates or inhibits transcription. Okay, so this right here is a diagram that shows how a specific gene is affected by a growth factor. In the reception stage, the growth factor.
Jack, could you point it out, like when you're actually...
The growth factor binds to the receptor. The receptor then relays a message through relay molecules that starts a phosphorylation cascade in the transduction stage, which is the transfer of the message. At the end of the transduction stage, there's an enzyme called a kinase, which transfers phosphate groups. The kinase then brings the message into the nucleus, which activates a transcription factor. The transcription factor then transcribes DNA into mRNA, which then synthesizes genes.
Now the transcription
Synthesis
factor, how does it do that? Are you able to go into that? Because I know RNA polymerase transcribes DNA into RNA, but what exactly does a transcription factor do?
Doesn't it make an exact copy of the...
Does anyone have an idea? The transcription factors normally bind to something inside the nucleus. It could be binding to the DNA or binding to the RNA polymerase to enhance or cause transcription to actually take place. That's what transcription factors do.
Okay, so the signaling pathway can regulate proteins, can cause protein synthesis, and regulation of proteins affects functioning proteins outside the nucleus due to ion channels and concentration gradients. So this right here is a diagram that shows how epinephrine breaks down glycogen. So now this experiment was led by Earl W. Sutherland in 1971. He is credited now what we know now as cell signaling. He pretty much started the whole process. Originally he had epinephrine, and he found out that it broke down glycogen, but he saw that it didn't enter the membrane, and so he tried to figure out the steps required. This pretty much just shows what happens in each step. As you can see, transduction, the signal is amplified between each protein. And then at the response stage, glycogen is finally broken down into glucose 1-phosphate, which then can be converted into glucose 6-phosphate, which is used as an intermediate for glycolysis.
Okay, so the fine-tuning of cell response, whether it occurs in the nucleus or the cytoplasm, it can be fine-tuned rather than being turned on or off completely. And what do you think could be the possible benefit of this fine-tuning, John?
It could moderate whether to allow this stuff to change it. If it's off, then no response can happen. But if it's on, then no response can happen.
Yeah, the cell signals, they can be amplified or decreased depending on the situation. So if you need more of something or less of something rather than it just being on or off. And one of the, there's 4 aspects of this, and one of this is cell response can be regulated at many different points. And this also is part of another aspect, which is a pathway with multi-steps could be regulated at more than one point. The third aspect is that the overall efficiency of the response is enhanced by scaffold proteins, which we'll talk about in a second. And then The crucial point is the termination of the signal. So that kind of determines how many steps there are in the signal and how many different places amplification can occur. And this is just an example of different amounts of substances with cell amplification and different responses. So here was a virus, and if there was an antigen that is weak or absent, and there's if there's a low T cell activity, which is the immune response, then the virus kills the host cell. Or if there's a medium or high, then the host cell is okay. And then if there's a moderate amount of virus and a high amount of T cell activity, then the host cell will die. And then if there's a strong amount of the virus and there's a high and a medium T cell activity, then the host cell will die too. So fine-tuning of cellular responses is aided by scaffolding proteins. And these proteins attach themselves to different parts of signal pathways in order to move them to the different areas of the cell. And here's an example of a scaffolding protein. And here we have the signaling components from the pathway. And they take on these different components in order to increase the efficiency and rate of cell response. And they also organize different parts of the cell response into what we know as complexes, and we'll talk about more of these later. All right, what do you think causes a cell response to act faster and in a larger amount? Sophia.
Is it that this protein, like you have more of them, so you have like more of the signal?
Yeah, but first, before even that, large amounts of enzymes cause this to occur, as the enzymes activate more proteins and make them stay active longer, creating more of a product. And as this occurs, the amount of products created in each step increases as the process goes on. Yeah, and then here, amplification is due to the large number of proteins that are able to create large amounts of product before becoming dormant again. And this amplification allows for cell response to be carried out with a much stronger response, which leads to an increased efficiency in cells, so it doesn't have to do the same process over and over again, rather it can just do it once while creating the same amount of product. And an example of this is epinephrine in the liver. And rather than using an insane amount of epinephrine to create a response in the liver, it can use just a small amount, and then the signal gets amplified as it travels through, creating a massive breakdown of glycogen and a release of glucose. And this is just the diagram of how it happens. Here's the epinephrine binding, and here's the G protein, which picks up the signal and starts transduction. And it activates adenylyl cyclase, and it activates these here, bringing the phosphorylase at the end step, and then glycogen is then broken down into glucose, which is then released into the blood.
Okay, specificity of cell signaling and coordination of the response. So think about a liver cell versus a muscle cell. Both are in contact with your bloodstream and therefore are constantly exposed to many different hormone molecules. Yet the liver responds to some signals and ignores others, and the same is true of the heart cell. Some kinds of signals trigger responses in both cells, but they are different responses. For example, epinephrine stimulates the liver to break down glycogen, but the main response of the heart to epinephrine is contraction, which leads to a more rapid heartbeat. The explanation for this specificity exhibited in cellular responses is because different kinds of cells turn on different sets of genes, different kinds of cells have different collections of proteins. The response of a particular cell to a signal depends on its particular collection of signal receptor proteins, relay proteins, and proteins needed to carry out the response. Thus, two cells that respond differently to the same signal differ in one or more of the proteins that handle and respond to the signal. Okay, now let's take a look at a visual representation of cell signaling. So in cells A, B, and C, they all use the same receptor protein for the red signaling molecule. And the differences in other proteins account for the differing cellular responses. In cell D, a different receptor protein is being used for the same signaling molecule, leading to a different response. In cell B, a pathway that is triggered by a single kind of signal diverges to produce two different responses. In cell C, two pathways triggered by separate signals converge to modulate a single response. And this branching of pathways, or crosstalk, which is another word for interaction between pathways, is important for regulating and coordinating a cell's response to information coming from different parts of the body.
Scaffolding proteins increase the efficiency of signal transduction by having all the protein kinases in one area. It doesn't restrict the efficiency of the signal to the diffusion of proteins through the cytosol. Because if you think about it, if each protein had to move through the cytosol after it's activated, it'd be like much slower. But if it's a scaffolding protein, it happens like that. Because they're on one area, it facilitates phosphorylation cascades. Scaffolding proteins contain different types of protein kinases, but they can also contain the same, such as the STE5 scaffolding protein in yeast, or that's the scaffolding protein. It contains STE11, STE7, and FUS3, and same as tyrosine kinase contains all of the same in the dimmer.
So in cell signaling, the termination of the signal is just as important as the actual activation mechanism, and it's essential for the pathway. And it's for the cell of a multicellular organism to remain capable of responding to incoming signals. Each molecular change can only last a short time, so it can be reversed. And that ability of it to react to different signals all depends on the reversibility of the changes that was made to the prior signals. And that's why the binding of signal molecules to receptors is reversible. And it's able to change as the external number of signal molecules falls so that fewer receptors are bound at any given time. And once that happens, it falls below a certain threshold so that it can revert back to its inactive form. And then an example of that would be the GTP phase activity intersynced to a G protein hydrolyzes its bound GTP, and the enzyme phosphodiesterase converts the C-AMP to AMP protein phosphates inactivate, and then phosphorylated kinases. So as a result, the cell is ready to respond to a new signal. And then this is the example of it. So you have the normal state, and once it's exchanged for GTP, it's active, so it leads to whatever that signal responded to, so it's growth and division. And once the GTP hydrolysis facilitated by the GAP, it goes back to its inactive state, so the growth is stopped, so it's reversed, and then it can get different signals.
All right, so apoptosis. So 50 to 70 million of your cells die every single day, but this is perfectly normal. This is part of apoptosis, which is known as programmed cell death, which you can see is occurring right here. And it's one of the most important and elaborate pathways of cell response. And this occurs in damaged and sick cells, or cells that have reached the end of their lifespan. And unlike necrosis, which is when your body is dying and all your cells are, the cell fragments are absorbed by neighboring cells, and so that other cells aren't harmed. An example of why apoptosis is so important, and these are cancer cells, and these are cells that weren't terminated and led to cancer, and that's a very terrible disease.
Canor habitus elegans, or as I'll abbreviate C. elegans from now on, is where we get our pretty much all our knowledge of apoptosis from. It's a very small soil worm. Here's a picture of it. It's only 1 millimeter in length. It only has 1,000 living cells, 1,000 cells total. So it was easy for researchers to track the entire ancestry of all their cells and see exactly which cells died, which cells stayed alive. They found out that apoptosis occurred exactly 131 times during this organism. While researching this, they revealed that there's two key apoptosis genes, CED3 and CED4. CED stands for cell death. These genes are always present in the organism, but they're always in their inactive form unless a death signal is received. Another important gene is CED9, which acts as the brake. It's always active unless a death signal comes in. Once a death signal comes in, it turns inactive, the brake goes away, cell dies. Let's talk about that. Alright, so here's a diagram of the C. elegans while CED9 is active. The CED9 protein, since it's active, it inhibits CED4 and CED3, so no apoptosis goes on. But once the cell receives a death signaling molecule, it turns CED9 to its inactive form, which activates CED4 and CED3. The activation of these two genes then produces proteases and nucleases. Proteases lead to protein metabolism. Nucleases destroy the phosphodiester bonds between nucleic acids, and thus those break. While the cell is dying, the cytosol forms blebs, which are like the protrusions that you can see in the cell when they're dying.